Okay, so for what we covered today, I am hopeful that the vast majority of this will be some level of review, okay? You guys should have gotten this in anatomy or your human physiology class, okay? I'm not going to require you to know like individual muscles, individual nerves, right? This is not anatomy class. But we're going to talk to some extent about the details of cellular structures inside of skeletal muscle. Some of those structures, though, I am going to want you to know, all right? We will learn about, like, what is a T-tubule and what is the sarcoplasmic reticulum, okay? What are actin and myosin, and what's the sliding filament theory? So we're going to cover this in a way that makes the most sense to me, okay? It may not to you guys, but this is how it makes sense to me. So I think I can do the best job teaching you this. We're going to do some structure first, okay? And then once we kind of have the ideas about what the structures are, then we'll talk about functions, okay? So we'll do some cellular anatomy, then we'll do some physiology and some actual function, okay? And we're going to do that for skeletal muscle, and we're also going to do that for the parts of the nervous system that are going to allow the brain through the spinal cord to connect to skeletal muscle so that your brain can tell your muscles what to do, when to do it, how much force to generate, go fast, go slow, those kinds of things, okay? So it's really, really important that you guys appreciate at the end of all of this that the way that the nervous system and skeletal muscle are structured play an absolutely key role in imparting function, okay? If your muscles and nervous system were not organized the way that they are, walking would be difficult. Running, jumping, lifting weights, right, typing, everything that you do when you move, and therefore any kind of exercise and physical activity would be difficult, or they would be different, all right? And so that's why I think it's so important that we try to understand all of the individual structures, how they fit together, and how these processes work, okay? Secondarily, once we learn that, okay, then we can talk about how do those structures and those functions change when we do exercise training, be it resistance training or aerobic training, or how are they going to change when you get old, or how do they change when you have a certain disease, okay? So we're going to kind of hit this first. It's going to be review, I hope, but then once we can feel more comfortable with kind of how this works, we can do the fun stuff, which is how's it going to change, what's good, what's bad, that kind of thing, okay? So just kind of hang in there with me on all of this, and hopefully we can get to some interesting things, okay? Some kind of very general learning objectives. We're going to learn the basic structure and organization of these things. So some basic cellular and kind of macro structure organization of muscles. We'll learn about the sliding filament theory of contractions. So how do these structures fit together, and how do muscles actually contract, okay? What is happening when I ask you to stand up and do a squat, okay? We'll learn about structure, location, and function of motor nerves and motor neurons, and how the brain is going to communicate with muscles to do things. Okay, so we're going to start with this, okay? For today and probably the next couple of lectures, we're going to focus exclusively on skeletal muscle, okay? Skeletal muscle. And so skeletal muscle is a little bit different than the other kinds of muscle in your body, which are going to be cardiac muscle and smooth muscle, okay? We will cover cardiac muscle in the cardiovascular system. We'll cover smooth muscle as well in the cardiovascular system, okay? So what makes skeletal muscle unique and a little bit different is that skeletal muscle has stripes in it, okay? Those stripes are called striations. So if you look at it under a microscope, okay, it's going to have this very characteristic like patterns of colors, light and dark, and light and dark. It's very, very highly organized, okay? Do you guys get to see like electron micrographs or anything? Like you guys look at muscle under under magnification or anything as part of anatomy class? We all just focus on like, here's the bicep, right, of this poor older lady or gentleman that, you know, was there. We cut out their bicep. Let's look at, you know, kind of how it stretches. Okay, I'll show you guys a couple of pictures, but it has these stripes. These striations are there because of the way the proteins that make up muscle are going to be structured and organized. Okay, striations are due to the pattern of actin and myosin and how they're arranged inside the muscle. Okay, all right. You have about 600 or so skeletal muscles throughout the body. You will need to know the names of exactly 0 of those muscles for my class. Okay. You guys want to know something that I probably shouldn't tell you guys, but I think is pretty funny? I've never had a class in anatomy. I've never taken anatomy in my life. I've taught anatomy and physiology, and I've never had a class in anatomy. Okay? If you ask me to name a particular muscle or a particular nerve that is not like your bicep or your tricep, right, or your quad, don't ask me about abductors and adductors and that shit. I know nothing. Okay? I don't want to minimize how important that is, but I've managed to get a PhD and make it through life without knowing all of these kinds of things. So just kind of an FYI. So some of you, or probably all of you, will know more about some of the names of the individual muscles than I do. So therefore, I will require you to know none of them. Okay. The big thing to keep in mind about skeletal muscle that's going to separate it in some ways from cardiac muscle and from smooth muscle is that it is what we call a voluntary muscle. Okay, it's a voluntary muscle. So if I ask Allie to stand up, see the fun part of names. Allie, can you stand up for us, please? I asked Allie to stand up. Okay. Now, Allie, will you sit back down? Thank you. Allie did that under conscious control. She chose to get up. Her brain said, use your legs, do whatever, stand up, then sit back down. Okay? Skeletal muscle has conscious control. That is because of the way the brain and the nervous system are going to connect to it, okay, which we're going to talk about. But that makes it different. If, for instance, right, I was to say, Mitchell, make your heart stop beating. Think you can do that? You have okay. Let's not go into some of those, but you cannot simply by thinking about it make your right. If I'm like, ooh, I can maybe make your heart rate go up a little bit, or maybe make it go down a little bit. You don't have a lot of voluntary control, right? Can you imagine if you had to think about making your heart beat every time it beat, right? Think about how many times your heart has beat in your entire life. That would be crazy. You do nothing else. Or you're like me, and you'd be walking along and you forget. And you just, down you go, heart attack, okay? That separates skeletal muscle from cardiac muscle and from smooth muscle, okay? And there are particular structures that skeletal muscle will have that cardiac and smooth muscle don't, that allow that to happen. And that's one of the really, really cool things about skeletal muscle is because it's voluntary, because you can control it, then we can actually teach it to be controlled in slightly different manner. You can learn how to do new things, okay, because of all of that. You can create these new motor patterns, and we'll talk about that next week, okay? All right. So we're going to spend most of our time from a structure standpoint on the individual cellular structures, but I do want you to appreciate a little bit of the bigger kind of whole muscle down structures in muscle, okay? Does anybody know what a Russian nesting doll is? Anybody know what a Russian nesting doll? Okay, Camilla? Yes, right? So the little dolls, they're all identical, right? You have a little one, and there's a bigger one that you put the little one into, and then another, and another, and another, and you end up with like this thing that's like this big, and then it's all full of these little identical things that get smaller and smaller. Okay, I think of the general organization and structure of skeletal muscle as a Russian nesting doll. Okay, you have what we typically think of as skeletal muscle. So you've got this whole muscle. Okay, For instance, here you have my bicep, okay? Whole muscle. It has this somewhat like tubular structure. It's encased in a membrane, right? There's some connective tissue that surrounds all of it, which I'm sure you guys dissected and cut through and named and penned and did a whole bunch of stuff, right? So it looks a certain way. Well, if you were to cut through the subcutaneous fat, cut through the epimysium, right? You're cutting through the fascia that's going to be there, and you look inside of my whole bicep, what you would notice is a collection of what look like mini biceps, of like mini muscles that are in there that are called fascicles, right? And those fascicles have their own connective tissue around them, right? So you've got a big tube made up of smaller tubes. And then if you cut the fascicle open, what you're going to see are even smaller tubes that are making up these somewhat larger tubes that are going to be fascicles, okay? The fibers have their own connective tissue around them. So they're going to have an organization of these things. So here we have whole muscle. Here are these individual fascicles that are in there, okay? Inside each fascicle is going to be an individual muscle fiber. Inside of each muscle fiber, there are going to be many of these even smaller tubes called myofibrils. The myofibrils are made up of not so much tubes, but little segments of tubes called sarcomeres. And then inside the sarcomeres, they're made up of these long filament or tube-like structures that are made up of proteins. This is actin and myosin. So you basically have strings or tubes inside of tubes, inside of tubes, inside of tubes, okay? And that's the organizational structure of this, right? This may seem strange. Why would we do this, all right? I'm hopeful that after we kind of get into function and turning things on and off, that you will appreciate why this kind of organizational structure is somewhat useful to us, okay? But we're going to talk about kind of what each of these little things does, and then it will hopefully make a little bit more sense, okay? All right. As we look at all of this, okay, we're not going to spend much time on the whole muscle. We're not going to spend really any time on a fascicle. We're going to spend the vast majority of our time talking about fibers and sarcomeres, and then the individual contractile proteins that make up those sarcomeres, okay? All right, so here is a cross-section, kind of a cartoon cross-section of one individual muscle fiber, okay? One individual fiber. If Helen let me, I could take a scalpel and I could cut a small little incision in her quad, okay? And I could reach down in there and I could pull out and tease out one individual muscle fiber. You can see it with the naked eye, okay? It might be a couple of centimeters long, okay? If you pull one out. Now, in theory, if you come over to Sarkis and you tolerate xylocaine and things, I can give you a little bit of local anesthetic and you sign some stuff, we could conceivably actually take a little piece of muscle out, okay? Called a muscle biopsy. I can pull out a fiber. So you can still see individual muscle fibers without magnification. The importance of a muscle fiber is that one muscle fiber is a single muscle cell, okay? So if you think about it in that way, if you can see a fiber with the naked eye, can you think of any other cells in your body that you can see a single cell with the naked eye? Right? There are these huge, giant cells, okay? And that's going to make them unique, and it's going to give them a little bit of they're going to have some different internal structures than regular cells, okay? So one fiber is one cell. Depending upon the size of your muscle, you may have hundreds to hundreds of thousands of individual muscle fibers in one muscle, okay? So if we look at all of this, like every other cell for the most part in your body, a muscle fiber is going to have a lipid bilayer membrane that surrounds it, okay? That surrounds it. So it has this cell membrane, okay? You guys talk about cell membranes in human phys, or you at least maybe talked about the function of cell membranes in whatever it's called now, intro to zoo or biology or something? Yes? Right? Hydrophilic, hydrophobic, right? The little lipids. tails, heads that look like a little sperm or something that are going to be there. They're going to be signaling like channels and proteins and things that are going to be interspersed. Just like almost every other cell, muscle fibers have that. We call the membrane that surrounds a muscle fiber, we call it the sarcolemma. We don't really call it like a plasma membrane or a cell membrane. It's called the sarcolemma, okay? Sarco is either Greek or Latin. I should probably look it up, but it means muscle. That's what it means. Sarco means muscle, okay? So it's the muscle membrane. That's what the sarcolemma is, okay? We will talk about the sarcolemma. It has some very specific and somewhat unique properties that are going to help impart voluntary function onto skeletal muscle. Okay, so we're going to come back to that a little bit. Connected to the sarcolemma, if you look along the sarcolemma, you're going to see that there are going to be tunnels kind of burrowing down from the sarcolemma down into the inside of the muscle fiber. Okay? These tunnels or tube-like structures are called transverse tubules, oftentimes shortened to be called a T-tubule. Okay? A T-tubule. The function of a T-tubule, which are going to be the gold structures that you see here and here, and they're going to be interspersed all throughout the cell, is to provide, okay, an ability to take a signal from the outside here on the sarcolemma and bring it down into the inside of the muscle fiber so that we can tell something to happen on the inside, which is going to cause things in the individual sarcomeres and the proteins to happen. Okay? So a T-tubule is a signaling structure. Right? It's a signaling structure. If we wanted to, I could make your T-tubules not function very well or not function in a normal manner simply by having you get up and exercise until you can't do anything else. Okay? We could ask Callie to stand up and do squats until she can't squat anymore. And at the very end, one of the reasons that she can't squat anymore is that her T-tubules don't function right. And we let her rest for about 30 seconds or a minute, and then her T-tubules go back to functioning correctly. Okay? When we get to aerobic exercise, we will talk about something called fatigue, right? An inability to keep contracting your muscles. One of the primary sources of fatigue is improper function in the T-tube. Okay? So again, there's going to be lots of connections between these individual structures and a person's ability to function and perform certain tasks or perform those tasks over and over again. And that's why I think it's important that we learn and understand these things. So connected to the T-tubules on the inside of the cell is going to be a network of storage sacs that are called the sarcoplasmic reticulum. Okay? So sarco, again, meaning muscle. And what happens is they're going to be these kind of sac-like or bag-like structures. They're going to surround all of the myofibrils, but they're also going to have these very close connections to the T-tubule. Okay? Does anybody remember there's one thing that is stored in the sarcoplasmic reticulum that's really, really important? Remember what that is? Calcium. Very good. Okay? Calcium. For our purposes in class, that's all you need to know about the sarcoplasmic reticulum. It is a storage site for calcium. The ability of the T-tubule to tell the sarcoplasmic reticulum to release that calcium is one of the key steps in telling your muscles to be able to contract, or telling them, depending upon how much calcium you release, telling those muscles how much force to generate in comparison to their max. Okay? So again, structure is going to help us with function. Other things that are going to be inside a muscle fiber, these little kind of green and yellow structures that you see, they're going to be interspersed around in here. Those are mitochondria. Okay? Mitochondria are where aerobic metabolism occurs. This is the place where we use oxygen and glucose or fat or something to make ATP. Okay? Probably on Tuesday of next week, you will learn, well, why do we need all that ATP as we contract our muscles. And then it will become hopefully very obvious why we want to have mitochondria around and in here to supply ATP. Okay? We're not going to talk very much about metabolism in this class. We're not going to talk about internal and external membranes. We're not going to talk about the folds in the membranes in the mitochondria. We're not going to talk about oxidative phosphorylation or the Krebs cycle or the electron transport chain or any of the things you probably have already learned. We're just going to assume the mitochondria exist and that they work properly. Okay? I will tell you right now that not all muscle fibers have the same amount of mitochondria. Okay? We may get to it today, but we'll probably for sure we'll get to it next Monday. It is Wednesday? Yeah, y'all are Monday, Wednesday. I get my days all confused. We will for sure get to it next Monday. Muscles come in different flavors, okay? Or muscle fibers come in different flavors, okay? We call them fiber types, okay? And those fiber types are going to be denoted by some differences in the myosin protein that's in there. It's going to be denoted by some differences in some other enzyme functions in them. And depending upon the type, some types may have more mitochondria in them than others. Additionally, the number of mitochondria in these muscle fibers is also very much going to be dependent upon how much do you exercise, okay? If you are very, very active, you get lots of stuff that requires the generation of ATP very often, okay? You're going to make more mitochondria. And all of your muscle fiber types will have more mitochondria. So that will become important a little bit later on, but that's where the mitochondria are going to be. So the last piece here is this structure in purple out here, and it's called the nucleus, okay? So the nucleus is what's going to contain the chromosomes and the DNA in our muscle fibers, like every other cell. What makes muscle fibers very, very unique in the cellular world in your body is that because they're so big, okay, skeletal muscle has not one, not two, not three, but many, many, many individual nuclei in them, right? The nucleus is going to be the place in the cell where new proteins are going to be put together. It's going to help signal to make new or more mitochondria or other things that the cell needs to function and survive. Okay? So because it's so big, one nucleus is not going to be able to provide an adequate amount of all of the things that that cell needs. And so that makes skeletal muscle very rare. It's a multi-nucleated cell. Also, it's of no importance for our class, but the nuclei in skeletal muscle are out around the edges. They're not in the middle. Okay? That also makes them unique. In a lot of cells, the nucleus is kind of down in the middle. The skeletal muscle, they're out on the edges, but there's multiple of them. Okay? Anybody have questions about that so far? Those are some of our primary structures or organelles that are going to exist in skeletal muscle that I want you guys to know. Okay? Any questions? Everybody's doing good? Good. It's 11:45, ladies. How's our caffeine flow doing? You getting there? All right. We'll go get another cup here in just a minute. Okay. Let me see if there's any of these things that I did not cover. Let me see. Yep. Thousands to millions fibers per muscle. One fiber has many nuclei. What you see here, this is an electron micrograph of an individual muscle fiber. So even though you can see it with the naked eye, they zapped it under the microscope and they stained it in a certain way. What you can see here, the ones that are in green are nuclei, and the ones that are this kind of blue, which may be hard to see in the back, muscle cells also have something called satellite cells, which under a microscope, they look just like a nuclei. A satellite cell is a muscle stem cell. Okay? Stem cells are cells that can, in many ways, become any other kind of cell. Right? They're uncommitted. Right? And so in skeletal muscle, because there's so much protein turnover and things that happen with physical activity or being sedentary, you keep some stem cells to help just kind of incorporate new fibers and things and make new ones right there on site because they turn over. Okay, we talked about T-tubules, we talked about the sarcoplasmic reticulum. Okay, now let's take one individual muscle fiber, and remember that that fiber has many, many smaller little kind of tubes that are inside of it. Those are called myofibrils, okay? So what I'm going to show you here is this is a cartoon depiction of one individual myofibril. Now, the myofibril itself is not that important, okay? The things that compose the myofibril are very important, okay? So myofibrils are one long tube in essence, and that tube is going to be made up of these individual structures called sarcomeres, okay? So in a myofibril, I have a sarcomere, then another sarcomere, and another sarcomere, and they're just going to line up like that, okay, to make our tube. And so sarcomeres, in turn, okay, are made up of protein. And so they're going to be, inside the sarcomere, there's going to be these filaments, okay, kind of strings, if you will, that are made up of proteins. And so we call those filaments either a thick filament or a thin filament, based upon what they look like under a microscope, okay? And so each kind of filament in a sarcomere is made up of a different protein. Thick filaments are made up of myosin, and thin filaments are made up of a protein called actin, okay? So muscles are, for the most part, protein and water, okay? When you eat food that has protein in it, when you take a protein supplement, okay, before or after a workout, the reason that you do is because muscles are mostly actin and myosin strung together, okay? If you want them to get bigger, we have to make more actin and myosin, therefore we need the amino acids available to help put those things together, okay? For this particular class, you guys don't have to know this, but in exercise phys you probably had to learn this in either anatomy or in physiology. Sarcomeres have a very distinct organization. You guys will note these things called Z-disc and an I-band and an A-band and an M-line and an H-zone and all of this. All that these areas are going to denote is in one sarcomere, what is the arrangement of actin and myosin relative to each other inside that sarcomere, okay? I'm not going to ask you, like, what's the M-line is the middle point, the Z-disc is the end point, okay? I'm not going to ask you guys about that. I'm not going to ask you, okay, in the M zone, what is the relationship of overlap between actin and myosin, okay? What I do want you to know, though, is roughly, in a very general sense, how are actin and myosin going to exist kind of next to each other inside a sarcomere. I'm going to show you guys some more pictures about that, okay? Before I show you the picture, I should probably say this. 2/3 of all the protein in a sarcomere, or really in a muscle in general, is myosin, okay? 2/3 of muscle protein is myosin. This is a good thing because myosin has a function as a molecular motor, okay? So myosin is going to be the protein, myosin is the structure that creates all movement that is generated by skeletal muscle, okay? So when you move, as you're writing, as you're typing, as you're breathing, whatever it is that you're doing, you're able to do that because your myosin interacts with actin in certain muscles, and the myosin moves. The myosin pulls the actin, and that creates movement, okay? Myosin generates force. It's a motor, right? Think of it like a car motor or something. Myosin generates force. So of all the things that are in muscles that are really important, myosin is arguably the single most important thing, okay? If each individual myosin molecule is able to generate a little bit of force, if I have more myosin, because I have a bigger muscle, I have the capacity to generate more force. Okay? If my nervous system knows how to communicate with my muscles in a better way, it can turn on more myosin, and I can get more force, or I can get that force in a very precise manner. Okay? So we need to really kind of worry about this. And so we'll talk a little bit more about the individual structure of myosin and some of its unique kinds of things. Okay? So here's a blow up of a single sarcomere. I apologize for you guys in the back. I know the yellow, that it's hard to see. Okay? One sarcomere runs from what's called a Z disc on this end to the Z disc on the other end. Okay? The Z disc it's a structure. It's a structural protein that's going to exist, and it serves as an anchor. Okay? It's like framing in a house, like in a wall or something. Okay? The M line, as I said, is this place right in the midpoint. The only important thing to know about the M line is that on either side of the M line, the myosin are going to pull in opposite directions. Okay? Myosin molecules are going to pull, as they change their shape, in towards the M line in every sarcomere. So in what you guys are looking at, the ones here on the left are going to pull from left to right. Okay? And try to slide these actin filaments in towards the M line. On the other side, on the right side, the myosin are going to pull from right to left. They're going to pull this direction in towards the M line. So what that creates is a scenario whereby when you turn on a sarcomere to get force, the sarcomere shrinks from both directions. Okay? It's going to shorten and squeeze in coming from both sides. Okay? So what you can see then is if we were to look at this under a microscope, we have these thick filaments that are made up of myosin. Okay? And a myosin filament is going to be this long string, and an individual myosin molecule, let me draw one. Okay? I cannot remember if I have a picture in the next slide or not, but an individual molecule of myosin looks like this, okay? It actually has two globular heads, but it's got a thing that kind of looks like this, okay? This back end of the myosin, this is called the tail region, and then this is called the head region up here, okay? And so to make a myosin filament, all of the tails of successive myosin are wrapped around each other, okay? They're going to be woven together. And then as you move along, what you're going to get is here, this tail is together, and I got a head going that way. And I got another myosin molecule here, its tail is woven in, but then its head is going to go off that way. And there's another one, and its head is going off like this, okay? And so what you get is these long filaments that in these very distinct little intervals are going to have these myosin, the heads of the myosin protein sticking off of them at these very precise angles, okay? And I'll show you guys a three-dimensional structure of that in just a second, okay? So they're going to look that way. Myosin is a bigger protein than actin. So when you look at it under a microscope, the myosin filaments look darker, okay? The actin filaments are smaller, their molecular weight is smaller, so they're going to appear lighter under the microscope. The alternating bands of these myosin filaments, actin filaments, myosin filaments, actin filaments, and these areas where out here towards the Z disc, right, there's only actin, no myosin. They're not overlapping, right? This area is going to be light. This area is going to be dark where you're getting both. And then this area is going to be somewhere in between here as we near the end line. The arrangement of these actin and myosin filaments create the striations in skeletal muscle. Okay? It creates this very distinct striping pattern because of how they're arranged next to each other. Okay? In a thin filament, which is this actin filament, Actin is a circular protein, okay? It's a little sphere, a little ball. And so you have two chains of these that are going to be wound around each other. So it's like one ball, another one, they make a big long chain that way, like you're stringing ping pong balls together or something. And then you take two of them and you just wind them around each other. That's an actin filament, okay? It's a little smaller, okay? It's going to weigh a little bit less than what's happening with the mice. Okay. Nope, I don't have a picture. Okay. You're not going to be responsible for this on the test, but I want to draw this and show it to you because I think it's going to make you understand why we want as much actin and myosin in our muscles as possible. Okay? It's difficult for me, or it's always been difficult for me, and maybe it's the same for you guys, to try to understand three-dimensionally what is happening inside of a muscle, right? You see these pictures, there are these 2D cartoons, look, here's one sarcomere, but these are 3D structures. And so imagine that we're going to take one, right, you've got a muscle fiber, inside that fiber, there are these individual myofibrils, we pull out one myofibril, it has these individual sarcomeres. So you're going to look down into one sarcomere. So you've got a sarcomere that's coming out of the whiteboard at you guys. Inside there, there's going to be multiple of these thick filaments. Okay, so you've got these filaments made up of myosin that are doing that. And then around them, are going to be the individual thin filaments. And I apologize, I've drawn this in a really terrible way. But what you get is there's a hexagon pattern of 6 actin filaments surrounding every myosin filament. Okay? And so the myosin has multiple heads. Some are pointing up to connect to this actin molecule, some are pointing down to connect to that one. But what this arrangement lets us do, because of this interaction, is we can take a sarcomere and we can pack it chock full in 3 dimensions of all of these filaments. It's not like there's one filament here and one over there. There's multiple actins around each myosin filament, and that fills up the entirety of the sarcomere. So it lets us get as much myosin and as many molecular motors in there as we possibly can. Okay? All right. Questions about any of that? Okay, let's take a break. Let's come back at 12:05. Okay, 12:05. Okay, I'm going to pick up again. So we are going to get to the fiber types today. I've forgotten what order I had to change the order of stuff just a little bit. Okay. So you guys talk about fiber types in like human phys or in anatomy? You guys are sort of nodding your heads. Okay. So I don't know how they teach them there, but for our purposes, I'm going to give you a little bit of the history behind how we realized there were different types of muscle fibers. They're not all the same, okay? And that there's a couple of different naming structures, both of which are right and both of which are not wrong, but maybe don't tell the whole and entire story about something, okay? So originally, okay, muscle fiber types were characterized as being either what we call fast twitch or slow twitch. Okay, you guys heard of fast and slow? That's probably how you may have learned, is there's fast twitch muscle fibers and there are slow twitch muscle fibers. Okay, I should probably give you some background on, okay, well, what does that mean? Like, how fast is fast? How slow is slow? Also, what the heck is a twitch, Dr. Black, right? What is a twitch? okay? As you all will see next week, when we want to turn a muscle on, we send from the nervous system, we send individual action potentials, these are little electrical signals, from the nervous system down into the muscle, and we tell it to turn on and for the myosin to bind to the actin and give us force. Okay? We'll go through all of that in excruciating detail next week. Okay? But for today, to understand a twitch, think of a twitch as one single individual contraction from that muscle fiber. Okay? A twitch would be what we get from one action potential and one action potential only. Okay? So to define something as slow twitch, what that means is that when we turn the muscle on, when we get that force from the one twitch, it's going to occur more slowly. So let me show you guys what I mean. So imagine here what we've got is we've got a graph, okay? Here's the 0 point, and what we're looking at is what we would call force or tension or something. It's the actual kind of workings of the myosin head, okay, where it binds to the actin and we get force. Here you are, you have a muscle, you're nice in there and you're relaxed, nothing is happening. Then I zap you with electricity and I give you one action potential, okay? You're going to get a twitch, and it's going to look something like where force goes up, and then it comes back down and it does this, okay? So, and we can measure what this is and say, okay, you generated 10 pounds of force or 5 pounds of force. We don't usually measure it in pounds, it's usually in newtons, but that doesn't make the least bit of difference, okay? Individual muscle fibers are going to have different shapes of this curve where it goes from being off to on and then back down off again. Okay? So let's assume that what you see here is a fast twitch muscle fiber. There's a certain amount of time that it takes to go from the onset of force to the top. Okay? If I have a slow twitch muscle or a slow twitch fiber, that time is going to be slower. And so it's going to look more like this. Okay? So what that means is I may get the same force, but it takes a little bit longer, it's slower to rise up to that peak force, and it's also going to be a little bit slower to relax back down. Okay? And so from initial experiments, mostly in animals, they cut a muscle out of an animal, they tease out an individual muscle fiber, they hook it up to these electrodes, and they zap it with one sort of action potentials level of electricity, and they were watching the shape of how it got turned on and generated tension. Okay? And what they figured out was some muscle fibers generate force very quickly, and some generate them a little bit less so, and there seemed to be a lot of similarity between like a population of fibers, okay? In a muscle, like there's a whole bunch of them that look exactly the same that are fast, and then a whole bunch that look exactly the same that are going to be slow, and then there's some that are kind of in the middle. That's where this idea of fast twitch and slow twitch muscle fibers have come from. It's about turning on the generation of force, okay? That is speed of contraction, so fast and slow, okay? You guys may have, in another class or in human phys, you may have learned that people that have lots of fast twitch muscle fibers are good sprinters, okay? They can run faster. That kind of makes sense, right? I can turn on the force faster. I'm a good sprinter. Whereas people that are more slow, have more slow twitch fibers, tend to be better endurance athletes, okay? Now, the endurance side is more subject to how much training they have, but in general, okay, you have more fast twitch, you can run faster, you can jump higher, things that require the application of force to take place very quickly, you're going to be naturally better at. Your skeletal muscle fiber type is genetically determined, okay? You have absolutely no control over it whatsoever, okay? Thank your parents or curse your parents, whatever it is, okay? It's genetically controlled, and it is going to be roughly similar. The distribution of how many fast ones and how many slow ones you have in each muscle in your body is going to be roughly similar, okay? Muscles that control posture, like your soleus, okay? Some muscles in your low back, which I don't know the names of, some of your abdominal muscles, okay? And then your diaphragm are going to have more slow twitch fibers. Because you use them more often, they need to be more resistant to fatiguing, so they're going to be more slow twitch, right? Your quads, okay? The muscles that control like your eyes, some of your the muscles that control your fingers, okay? Your gastroc, these are going to have relatively more fast twitch fibers relative to some other muscles in your body. The vast majority of you guys, if we took a biopsy and we examined your fibers, are going to be somewhere between 60%, 40% fast versus slow, one way or the other. I would say 3/4 of you guys are going to be somewhere in that range, okay? We might have one of you in here that is over 80% fast twitch or over 80% slow twitch, okay? Most people are somewhere with a pretty kind of even mix of fast and slow, okay? The folks that are on the very, very far ends, that's why not everybody's an Olympic sprinter, or everybody's not an Olympic distance runner, okay? Or it's why everybody's not even like a reasonable middle school sprinter or middle school like age kind of distance runner, okay? Most of us are somewhere in the middle, okay? I can take a cheek swab and see what kind of genes you have from all of that and approximate what your fiber type is. I can have you do a vertical jump or do kind of a sprint thing, and I can do it. I can bring you to my lab, put electrodes on you like a big TENS unit, and zap you with like 1 action potential, look at the kind of what the shape of your force tracing is, and kind of give you a rough estimate compared to everybody else in class. Are you more fast or are you more slow? Okay? So initially it was fast and slow. And this came out of animals and we took it into humans. Okay? What we know now is that that's not the entire story. Okay? People were very interested to know why. What is it that is different inside the fibers that makes some of them generate force faster and some generate force slower? What we know now is that this fast versus slow is dependent upon an enzyme that exists in the head of the myosin molecule, okay? And what we're looking at, that enzyme is controlled by, we're calling this, what here it says MHC. The MHC stands for myosin heavy chain. So on the myosin head, there's an individual section, okay, that is called the heavy chain. Yes, there is a light chain. It's not a medium chain, but there's a heavy chain and a light chain. And it's just a structure. Connected on the heavy chain is going to be an enzyme called the myosin ATPase. Okay, myosin ATPase. Fast versus slow depends upon the activity of that myosin ATPase enzyme. So if you have lots of fast twitch fibers, the myosin ATPase works faster, which means that myosin can generate force and then shut it off faster than a slower myosin ATPase, which makes for a slow twitch fiber. Okay? And so it's that ATPase enzyme, which is coded by that myosin heavy chain, that's the thing that's genetically determined. Okay? So by looking at, using molecular biochemistry, looking at the myosin heavy chains, they come in different flavors, okay? And so we call the heavy chain type 1, type 2A, or type 2X. There's actually some intermediates that exist between 2A and 2X. There's also a 2B, which tends to occur in animals, okay? But if you look at it this way, type 1 heavy chain is a slow twitch fiber, okay? So I will sometimes use those interchangeably. I may call it a slow twitch fiber, or I may call it a type 1 fiber, right? Those are the same. So type 2 heavy chain, those are fast twitch. So there is variation in the extent to which a fast twitch fiber is fast, if that makes sense, okay? Type 2X are the fastest human fiber types. Type 2A are not quite as fast, but they're still faster than slow twitch. Okay? When we get into aerobic training and resistance training, based upon activity, there can be some switching around of 2A and 2X fibers, okay? And they're going to have slightly different metabolic profiles and slightly different force generating profiles. Sometimes people also want to try to type your fibers based upon their metabolic activity. So to what extent are they oxidative? So to what extent can they make ATP through oxygen, which just means basically how many mitochondria do they have. Slow twitch fibers, okay, type 1 fibers have lots of mitochondria. They can make lots of ATP in aerobic metabolism, which is why they're good for endurance. Type 2X fibers, this brand of fast twitch fibers, have almost no mitochondria. You contract the 2X fiber, you get like 2 contractions and it's worn out. It needs a break, okay? The type 2A fibers, these kinds of fast twitch fibers, it's a little more complicated, okay? They're your friends that don't really know what they are. It's complicated, okay? So in the 2As, they might have a lot of mitochondria or they might not. It just depends upon how active you are, okay? So there's some level of trainability in the number of mitochondria. But that's what our fiber types are going to do. Fast twitch, slow twitch. Okay? Questions about that? Anybody has questions? Do we all need to like stand up and see who can jump and touch the ceiling and then guess it who's a fast twitcher and who's a slow twitcher? Or anything else? No? Any of you think, I can't get the ceiling? There's no way. Not in my teens, certainly. No. They are my stretchy jeans, though. This is a total side note, like the fact that they make men's jeans that are all stretchy now. Ladies, y'all been hiding this like stretchy fabric for years. The last couple where the men's are made out of stretchy things, oh, it's the best. I have dress pants that are like wearing leggings. They don't fit that tight. Nobody needs to see like that much like man thigh. But they're stretchy. My wife laughs at me. She's like, "Oh, women's pants have been like this for years." I'm like, "Y'all have been holding out on us." Okay, so we talked about this. Yep. You kind of hit this. Here's a table, okay, that's going to, in general, break down kind of the ideas of what may be different between these. Some of this stuff we won't talk about until later on in the semester, like how many capillaries those fibers have around them, how they tend to store fuel, are they storing fat, are they storing carbohydrate, okay? But that's kind of some of the stuff if you want to kind of get after that, okay? I hope maybe if you're looking at the study guide, it would be very easy, right, for you guys to envision a test question that says something like, compare and contrast the individual muscle fiber types, right? Talk about them as it relates to speed of contraction. Talk about them as it relates to their number of mitochondria, et cetera, okay? I'm not saying I'm going to ask you that question, but I'm not saying that I'm not going to ask you that question. Okay. I'm going to do. All right. So a couple of other points on fiber types, and then we're gonna switch over and talk about the nervous system side of things. Okay. So I told you guys that genetics determines what your fiber types are. That part is true. Weirdly though, despite your genetics, I can switch your fiber types around if you will let me cut the nerves that connect to those individual fibers. Okay? I'm guessing you guys don't talk about embryology or much of kind of growth and development and like the stages of all of that in anatomy or in phys or anything, probably. There probably is a class in biology that's like embryology or kind of those sorts of things. But what we know, and this is very strange, is that it seems to be that the nervous system determines the fiber type of an individual fiber. Okay? So, as you guys will see in the next few slides, in your spinal cord, you have a bunch of nerve cells that are called motor neurons. These motor neurons are going to send off what you think of as a nerve, which is really an axon, that goes from the spinal cord out, and then it's going to connect to each individual muscle fiber. Okay? Every motor neuron is going to connect to hundreds or thousands of individual muscle fibers in the same muscle. If I cut the axon from the motor neuron going to a certain population of fibers, okay, they were slow twitch fibers. I cut that axon. I have next to that, I have an axon that's going to fast twitch fibers. I cut that axon, then I go in and I swap them, okay? I connect what were slow twitch fibers to a motor neuron that was innervating fast twitch fibers, and magically, it turns those slow twitch fibers into fast twitch fibers. Or if you do the opposite, if you cut the nerve and you connect fast fibers to a slow motor neuron, then they become slow. Okay? So the genetics, the genetic potential to be either fast or slow seems to be there in every fiber. But which one gets turned on seems to be determined by the motor neuron. What determines the motor neuron, that seems to also be genetic, obviously. So in addition to having slow and fast fibers, you're also going to have what we might think of as slow and fast motor neurons. Okay? And the neurons turn on and off just at the same speed. They're not actually slow or fast, but they're going to make the fibers that way. Okay? So there's some really fascinating animal experiments where they cut axons and they rearrange them and they swap fiber types around. Where this really matters in people, okay? And it really matters in people is with aging. As you get older, some of your motor neurons are going to die. And you're going to lose connection to a certain population of muscle fibers. For whatever reason, the neurons that tend to die are almost exclusively neurons that connect to fast-twitch muscle fibers. And so those neurons die, your existing, still functioning neurons sprout off new axons to try to reconnect to those muscle fibers so you can have control over them. And so as you age, you tend to lose fast-twitch fibers because you've got more and more axons coming from slow motor neurons that are connected. And that's partially why old people are slow, right? They can't run as fast. The reaction time goes down, right? They can't drive because they can't sprint in those kinds of things. So that's going to be there. Okay. We talked about that endurance training may, in some ways, will shift 2X fibers to be 2A fibers. To be honest, it's not endurance training. Any kind of muscle contractions, resistance training, endurance training, just simply being active, the body does not like to have type 2X fibers. They're very, very metabolically inefficient, okay? And so if you do anything, any kind of activity, you're going to drive your 2X's to be 2As, okay? So it's not changing your actual fiber type percentage, it's just changing the flavor of your type 2s, okay? The converse happens when you sit on your ass. When you're inactive, your A's become X's. So it exacerbates this idea of eating. Okay. Questions about any of that? Feel like I'm beginning to lose you guys a little bit? Do we need to do some squats? Do squats until somebody answers a question? Or you want, we'll just stop here. This is fine. This is as good a place as any for us to stop before we get into motor neurons. Okay? So we'll stop here. All right? There is no assignment for Monday. All right? No assignment. Have a good weekend. All right? We'll just cut this off and we'll pick it back up then.